Hello everyone, today we will learn about the basics of Biovia Discovery Studio, which is a visualizer tool and mostly used for the visualizing molecular interaction and analysis of binding mechanism. So this is basically open as a software that you can directly download from discover.3ds.com by using your email ID, and after successfully register you can see this page where you can directly download the exe file of the application according to your operating system requirement. Once the application is downloaded and installed on your desktop, you can directly open it from your desktop screen. Now this is the window of the Discovery Studio Visualizer. Now we'll open the 3D structure of the protein. So this is the 3D structure of human hemoglobin that you can easily download from Protein Data Bank, and to move or to operate this structure you can use the rotate tool or the translate tool. By using the rotate tool you can rotate or move the structure to any direction, and for better clarity you can also change the background color. Before any kind of interaction and analysis, if you zoom in the structure, you can see there are few water molecules, and these water molecules need to be removed from the structure. So to remove the water molecules, you can go to the script, selection, select water molecules and delete. Now we have the optimized and refined structure of human hemoglobin. You can also see the structure along with its sequence. So click on sequence, then go to the source sequence. It will give you the information about the entire sequence of the structure. And if you highlight the part of this sequence, part of the region also will be highlighted within the structure. Now this structure has 4 heme group as its ligands, and to know more about the ligand binding sites and how it is interacted within the structure, we can click on the script, go to the ligand interaction, then click on show ligand binding site. This option will give you the binding information or how the heme groups are being attached to the structure. So these are the 4 binding sites that present within a structure, and in the side views we have the ligand interaction. So by clicking on ligand interaction, it will give you the enlarged version of one of the heme group. Now these are the residues that binding to the heme group, and these residues can be also labeled and the view can be changed to any other form. So I'm changing the view to the ball and stick model. You can also add the label to the residues. Change the atom to the residues. You can also change the font size. Now here you can see the residues are being labeled within the structure. We have also different options like show distance and show type. Here we can know the distance of the bond and the type of the bonds that form between the residues. By clicking on show distance and show type, it will give you the information about the type of bonds and what is the distance between the two residues. You can also spin the structure for better view. We have also another option called 2D diagram. So these are the two different ways to visualize the structure. One is 3D view, another is 2D view. We have also other surface option, like you can see the or check the hydrophobicity and the hydrogen bond region of the structure. Here you can check the hydrophobicity region. You can also have the scale and measure the hydrophobicity of the structure. And by clicking on hydrogen bond, it will differentiate between the donor and acceptor region of the structure. You can also expand the region to know more about the hydrophobicity and hydrogen bond donor and acceptor region. To know more about the binding mechanism or detail about the binding mechanism, you can go to the chemistry, click on calculate basic properties. This option will give you all the information about the binding or all the other parameters that involved in the binding. For example, if you clicking on amino acid chain, it will show you the secondary structure along with the binding site region. Similarly, if you click on the non-bond option, it will give you the information about the type of bond, what is the nature of bond between the residues, and what is the partial charges. Also, you can check the charge of the structure. So these are the basics of Discovery Studio. Thank you for watching.